Okay, so I'm known for the resources that I create for my courses, but I get more positive feedback on these audio test reviews than anything else. So since this is your first one, just kind of walk you through a few things. Number one, this is a part of the learning process. I want you to try to answer each of these questions on your own, and then if you can't answer them, find the answer. And that can include texting me, or obviously you should go to your book and go to your resources first. So hunting down the truth and hunting down answers is very, very important. But once you do that, come through here, and then I will show you how I think about things. And of course, I make your tests, and I know what's important based on learning objectives and those kind of things. So you should have this review basically filled out, except for maybe a couple tricky spots or these kind of things. And then listen to this recording and change your answers and add to your answers, and come to me if it leads to more questions. That's totally cool. So this should not be something you do right away. But at the same time, it may benefit you to listen to it once early on, as a basically a form of lecture, and then listen to it again or several times as you're getting ready for each exam. So that's kind of how I use it. So why do I make such detailed reviews? Just so you know, not everything on the test will be on your reviews. Not everything on the review will be on the test. I make the unit reviews, right? What are the things that I think are critically important about a unit? And then I make tests based on those things. But I don't ask a question about everything that's important. The tests would be hundreds of questions long. So they're really unit reviews. They're designed to make sure you know all the key stuff from the unit, or most of the key stuff from the unit. So what I like to say is that I try to make a test so that if I looked at this review, I could say, well, if they understood each of these things and not just regurgitate this one word or something, if they understood all the things on this, they should get a B on the test. That's what I shoot for. So my job is a filter. There are teachers that don't like test reviews, but my job is a filter. My job is to take your textbook. Let's see, I got it sitting by me. Let's take a look here. You got a textbook that's about 600 pages long. My job is to filter it to what's in reality, if you learn 25 or 30% of what's in this book and what's in this course, and you remember it, consolidate into a long-term memories, I'd be ecstatic with that. When I teach a human biology or human anatomy class, most people, if you're going to go to medical school, you're going to take anatomy 5 times. I spent a year or more, a year to 2 years, learning each organ system. So what I teach you in way less than a week, if I teach you in like 2 days, I would have spent a year learning. So my job is to determine what's the most critically important stuff so that I can keep my promises to you based on what's in the syllabus and what's important in this class, and that you are ready to move on for whatever you're using the class for. So all right, let's dive in. So the Unit 1 test review. Unit 1 is over chapters 1, 2, 3, and 4. So chapter 1. What is science and what is biology? So the science is the study of nature, the study of the natural world. I like to say that it's the objective search for truth, where we use the scientific method to follow the truth wherever it leads. That's what I like to say. But it really is just the study of the natural world, and that's the key. So what isn't science? Science can't observe, test, and repeat everything. Science has issues with too many confounding variables, as far as teasing out what the actual truth is. And science has no bearing on the supernatural, or it only deals with the natural world. So then what is biology? Biology is a subset of science where you study life, study living things. Whereas like chemistry is a study of matter and energy, and biology is a study of life. So, and then of course there is biochemistry, where we have to understand chemistry in certain areas. That's what, which we'll talk about in chapter 2 and moving on forward to understand biology as well. So we can't completely divorce biology and chemistry, but that's what biology is, study of life. All right, what are some of the key characteristics that allow something to be classified as alive? So the main one to me is maintaining homeostasis. In an ever-changing world, a living thing can maintain a stable internal environment. We have the ability to grow, ability to reproduce, having a metabolism. These are all things that would allow something to be classified as being alive. So to me, growth, reproduction, responding to the world around you is a big one. Maintaining homeostasis, these kind of things. All right, so that's how you classify things as being alive. So the difference between me and the table here, I don't even know what this table's made of, but is, I have a metabolism, I can maintain homeostasis, I'm alive, and it's not. So, all right, next, what is homeostasis and why does it matter? So homeostasis is the maintenance of a stable internal environment. That's the sterile contract definition. But what it is it's really hard to be alive. And life, at every second of the day, things are happening to you that could make you fall into a state of disease or death. Homeostasis keeps that from happening. If if your pH is changing in your body will respond to try to fix it. If your body temperature is going up, your body will do things to bring it back down. These are examples of maintaining homeostasis. And so why does it matter? Just because of that. We live in an ever-changing world. And look, you have cold-blooded animals can't live at extreme temperatures like we can. We can do things other animals can't that have allowed us to dominate this entire planet. So we've needed changes. We'll learn later on. We've needed changes to our skin color, etc., as we've moved farther from the equator. But the fact that we can maintain homeostasis, really like no other species, is the reason that we have taken over this planet. Now, we'll talk later on of whether or not that's a good thing and how many humans there should be, etc. But that's why it matters. It is so much easier to be dead than alive. And if you can't maintain homeostasis, you're not going to be alive. Now, forever, right? I mean, for long. Like I say, you have pre-diabetes. You're not going to die from pre-diabetes. But there are 100 million Americans right now that have diabetes or pre-diabetes. And pre-diabetes means that you have an impaired glucose tolerance. When you consume a lot of glucose, your blood sugar levels get higher than they should, and they stay higher than they should. So it's not going to kill you, but you are in a state. There are abnormal lab values. You are not perfectly healthy, and the average pre-diabetic within 5 years will be a diabetic, and the average diabetic will live, I don't know, somewhere in the neighborhood of a shorter 8 years shorter life because of it. So yeah, that will kill you. It's just a matter of when. All right, what are some of the key features that set humans apart from other species? So this would be, to me, we've got the opposable thumb. There are other species that have it, but not many. The opposable thumb, which allows us to grip things and use tools in ways other animals can't. I don't have to be as strong as a rhino because I can hold a gun and things like that. I don't have to be... we don't have to fly as a species because we learned how to, we created airplanes and these kind of things, right? So the opposable thumb is important, and then the massive frontal lobe. We have a huge brain relative to our body size, and it is massive, and especially our frontal lobe, which allows us to do planning and to predict the future, right? Understand the outcomes of our actions, these kind of things. Most animals are just driven purely by instinct, and we have those instincts, but we also understand when we can override them with our massive brain. So those are the two biggest ones. We can regulate body temperature better than any other species, really. So opposable thumb, large brain, those really are the big ones. All right, know the levels or hierarchy of organization from least to most complex in living things, as well as the world around us. I'm actually going to go to a picture of that so I don't forget any of them. So we have atoms and molecules. So atoms are the basic unit of matter. So all things that are alive are certainly made of matter. And so atoms and molecules are going to build cells. So the second level would be cells. I would put between them the cell organelles, the fact that your cells are made of their own miniature organs that have divisions of labor and power cells. But you go from atoms and molecules to cells. So the cell is the basic unit of life. It's the smallest unit of something that's alive. When you put a group of cells together that have a specific function, that's going to be a tissue. So cells make tissues. We're just going up in levels. Then when you put two or more tissues together that have a shared goal, shared functions, that makes an organ. When you put all the organs and accessory structures together that are working together, that's an organ system. Then when you put all the organ systems together, it's an organism. Let's go back. Let's think like a heart muscle cell. So the cell is a cardiac muscle cell. You put all the heart muscle cells together, that's the heart muscle tissue. But it's not the heart, because the heart has a conducting system made of nerves. It has a fibrous skeleton. So you put all those together, heart muscle cells, heart nerve cells, and heart cartilage and fibrous skeleton cells. You put all those together, we now have a heart. Well, your heart works with your arteries and veins and capillaries, your blood vessels, to create the cardiovascular system, and of course blood as well. Then when you put the cardiovascular system along with all the other, total of 11 organ systems, you now have an organism, a human. So that's the level of organizations in living things, and that's one example. Then we have populations versus communities. A population is all the members of one species in a given area. Community is all the members of all species in a given area. So one human organism is part of the human population. Then the human population is the part of a larger community, which incorporates all living things. Then we go to the ecosystem level. That's the living and the non-living things in a given area. So that's the difference between a community and an ecosystem. Is ecosystem also has the non-living things. And the highest level of organization is the biosphere. And that's when you put all ecosystems together. Okay, Next, another big one, understand each step in the scientific method and the role that it plays in our objective search for truth. I put objective in quotation marks because I've already told you that a big problem with science is the fact that we're not always objective. Like it's hard to get someone to change their opinion or mind about something when their paycheck demands that they don't. The reality is I love my job and I want to teach you guys the truth, but my family is my priority and taking care of them. If somebody came to me and said, hey, would you come work for us? We'll pay you 3 times as much. And I don't care about money, but I care about security for my family. We'll pay for all 3 of your kids to go to Ivy League school. But you've got to, be less flexible, I guess, with the truth. That terrifies me that I would say yes to that, but I think I would. So I wish I could say that I would make a stand there, but I don't know. So the reality is that science is not as objective as we want it to be. If scientists were all robots, and someday scientists will all be robots probably, then that might be different. But so the steps. So first is just observation. I like to use the word ponder, just pondering the world around you. Then you formulate your hypothesis, which is an educated guess. So, you're observing the natural world and making an educated guess as to why you think it is that way. And then you're going to make a prediction. So this would be I call them if-then predictions. If my hypothesis is true, then this is what will happen when we test it. Then you're going to test it with an experiment. And we talked in the lecture video about what makes a good experiment. And we can talk about some of it here too. But if your experiment ends up proving your hypothesis, or you can't prove a hypothesis, but ends up showing that your prediction was correct based on your hypothesis, then you're done. Then you try to get it published in peer-reviewed literature, and you get the information out there for other people to look at it and test it themselves. If not, you don't give up. You don't go back to the beginning. You go back to the step where you formulate a hypothesis and you refine your hypothesis, and you do this again and again. And then once you've done that, then your hypothesis has been as close to proven as possible. All right, what is a control and why are they important? So a control is a standard of comparison. If you want to prove that a drug does this and causes this side effect, well, you got to compare it to something. That's what a control group would be. Like in my microbiology labs, we use bacteria as controls when we do genetic modification experiments. So if we're going to genetically modify E. coli and see how it behaves, then I want to see how regular E. coli behaves. That's going to be our control. So a standard of comparison. Without a control, you want to see the data is just not that valid. So because think about if I'm selling a blood pressure pill and I say when people take this pill, their blood pressure drops 10 points. Well, and you didn't use any controls, the reality is, especially with antidepressants and blood pressure medication, the placebo effect is really powerful. Placebo effect can be up to 30% of the improvement you see. So if people are given a fake pill, their blood pressure might drop 7 points. So I can't say that the drug is responsible for a 10-point drop in blood pressure. I can only say that it drops it 3 points lower than nothing, than a placebo, right? So that's why controls are really important. Okay, what is the placebo effect? It is the fact that when you think, our minds are very powerful. There's a book called Molecules of Emotion that goes into that. It's pretty old, but it's still worth reading. If you believe something is going to happen, right? If you believe something is going to work, then you're probably right. Like when people get diagnosed with cancer, if they believe they're going to win the war against cancer, the battle against cancer, they are more likely to succeed than if they don't and they give up. My wife sees people, she works in digestive oncology, and she sees people, they give them months to live and they live weeks because they've given up. And I'm not, that's not what I'm talking about here, but the reality is our minds are very powerful. If you think a drug's going to help you, it's more likely to help you. But it's not even that. Studies have shown that like if I give you 2 placebo pills, it's more effective than 1. If I give you a color placebo pill, it's more effective than a white one. If I give you a placebo gel cap, it's more effective than a tablet. Like we can truly be tricked. You can give yourself a placebo and still see some improvements. That's crazy to me. So the placebo effect is very powerful, so we have to account for that. It's like I just mentioned with the blood pressure medication. That's another. There's also the nocebo effect. If you think something isn't going to work, or you think you're going to get side effects, you're also more likely for that to happen too. So that's another piece of the puzzle. So just the placebo effect is, it just shows you how powerful the mind is. Just thinking something will work and help. And there are times that this is probably my favorite example. If you explain what you're going to do to someone during surgery, they're more likely to have a better recovery. They should have no impact on what happens during surgery and during the recovery process, but understanding the procedure and trusting the doctor and feeling like it's going to work helps them recover from surgery better. So, all right. What's the best number of variables to test in a quality scientific experiment? It is 1. You want the control group and the test group to be almost identical, except for the one thing you're testing. What's the problem with having too many variables? Then again, we don't know if someone, and I'm not saying this is bad. If you want to get healthy, do everything you can. But if you're trying to prove how effective exercising is for better health, well, if you tell someone to exercise and drink more water and get more sleep and eat better, and they do all those things and they get better, we can't say how much of it was the exercise. So that's why you want the number of variables to be 1. If you see a difference between your control group and your treatment or test group, you want it to be because of the variable you were testing. So what's a confounding variable? Again, someone gets better, someone gets worse. A confounding variable can be something that we can't say why they got better or worse because there's too many variables. Let me give you a big example from the Iowa Nurses Health Study. This is where, in the 90s and early 2000s, people were doing a lot of hormone replacement therapy. And there are people still doing it, but for different types and stuff. But this is back when they were just giving women Premarin and all sorts of these medications constantly, and just for anything really, to help battle the symptoms of menopause, etc. But the reason they were doing so is because this Iowa Nurses Health Study found that the nurses that were using these drugs were healthier and had less heart disease than people that didn't. But the problem is they found later that when they actually tested that, they found that when you take those drugs, it increases your risk for heart disease. It improves bone health and symptoms of menopause, but it increases your risk of heart disease and greatly increases your risk of cancer. If you took Premarin for 10 years, you had like an 80% chance of getting uterine or ovarian cancer. Terrifying stuff. The largest study ever done was supposed to be a 5-year study. They stopped at 3 years in because they couldn't morally or ethically keep people on these drugs. They were doing so many bad things to them. So what happened? The hormone replacement therapy was a confounding variable. They found that the women that were, what they had in common wasn't the drug, is they were proactive about their health. They drank more water. They were less likely to smoke, more likely to exercise. They got physical exams on a regular basis. They were an active participant in their health. They were doing so many things that were good for their health that they were healthier in spite of the medication they were on, not because of it. So that's a great example of a confounding variable. All right. What is a hypothesis? What is a theory? And what is the difference? So a hypothesis is an educated guess. Your best guess at trying to explain a natural phenomenon. A theory is if an hypothesis has been tested and observed and tested and repeated and run through the ringer, then and we think it's true. How I define a theory is something with the highest probability of not being wrong. So if it's been very reliable and been very well tested, then the goal of a hypothesis when it grows up is to become a theory. So that's the difference. Anyone can formulate a hypothesis. An hypothesis can't be proven. Like observational research, or observational data, you can formulate good hypotheses. You cannot answer a question with a hypothesis. An hypothesis has to become a theory before we actually give it much weight. And even then, we have to understand that that theory can be disproven tomorrow. Okay, what is peer review and why is it important in science? So peer review means that when you get your data published after you've gone through the scientific method, that a group of your peers in your fields are supposed to look at this and call BS on it if there's something wrong with it before it ever gets published. So you should have more faith. If something's been published in a peer-reviewed journal, you should have more faith in it. That's why it's important. So making sure that, because anyone can say anything. But having a group of your peers say, no, this seems legit, that's a really big deal. It's like a stamp of approval. All right. What is a blind study or clinical trial, and what does it mean if it's double-blinded? So in a blind study, we talked about the placebo effect earlier. You've got a group. The placebo effect is going to be more powerful if people don't know it's a placebo. The crazy thing, though, is that even if they know it's a placebo, it'll have an impact. But if you're in a drug trial, a blind study or blind trial, you get a pill. You don't know if it's the treatment or the placebo. But the doctor does. The person giving you the pill does. In a double-blind trial, the doctor doesn't even know. Somebody else does. It's on a chart somewhere. But so the doctor and the patient, the experimenter and the experimental group don't know the truth. That's what double-blind means. That's important because we can be biased. I was involved in a clinical trial on chronic neck pain, comparing medication, acupuncture, chiropractic, these kind of things. Well, if I had a horse in the race and I wanted one of them to win, and I knew that you were the one getting acupuncture, and I wanted to prove that acupuncture was effective, I would say, the range of motion does look a little better, or you do seem a little better, right? But this was a double-blinded trials, so okay, sorry. So what I was talking about was, so if it's double-blinded, I was testing people, but I didn't know what treatment they had. So if I was falsifying the data, I wouldn't know which group I'd be helping. So that's why a double blind is good. It's to try to combat the fact that we have worldviews and we're subjective. Okay, what is the relationship between antibiotic use and the development of antibiotic-resistant bacteria? Did the use of antibiotics by humans create antibiotic resistance or just make it worse? So, all right, here's how I look at antibiotics, and I teach microbiology. Every antibiotic has a counter. It has a number of times it's going to work before it becomes useless. And every time it gets used, that counter is going towards zero. Now, I don't know what the number started with, but I know what's going to happen when it gets to zero. The drug's going to be worthless. Like methicillin, you've probably all heard of MRSA, methicillin-resistant Staphylococcus aureus, or MRSA infections, right? Staph infections. Methicillin isn't even used anymore. The counter has reached zero. Too many organisms have evolved resistance to it. So every single time we use antibiotics, we kill organisms that aren't resistant to it, but we don't kill the ones that are. So like if you look at a can of Lysol or something and it says that it kills 99.9% of germs, well, that's great. But what it does is it actually forces that 0.1% it can't kill, now has more space and more food, more real estate. It actually forces evolution forward. So when we use antibiotics, we are destroying their curative powers. I'm not saying not to use them, of course, but we can't abuse them, misuse them, or overuse them. If we're using them, we're wasting them, right? If we're using them to fatten up animals, or we're using them to fight viral infections, or we're using them as part of fertility treatments, all things that happen, we're destroying their curative powers by overusing them or abusing them. So it's just like evolution is a species adapting to their environment. If bacteria have to adapt to an environment full of antibiotics, the ones that can resist those antibiotics are going to become more common. That clearly happens. All right, did the use of antibiotics by humans create the resistance or make it worse? It actually didn't create it. It made it worse. So they've tested the microbiomes, the gut bacteria of people that live in the rainforest and have never come into contact with antibiotics or civilization, if you want to use that word. Probably not a good word, but and they already have organisms with antibiotic resistance genes. And that's because we didn't make antibiotics. We found them. Penicillin comes from the Penicillium mold, right? So the definition of an antibiotic is it's made by one microbe to inhibit the growth or kill other microbes. So microbes are at war with each other. The mold Penicillium has been fighting staph as long as the two have existed. So we concentrated it, made bigger doses and put it in pills. But we didn't start the war. They were already at war with each other. So they're fighting an evolutionary arms race, and we jumped in. But no, we did not create it. We created the problem. We made it much more beneficial to be antibiotic resistant. But we didn't actually create the antibiotic resistance genes. We just selected for them, is a term you'll learn later in the semester. All right, what are the key limits of science? True science with a capital S, which I should have capitalized here, I guess, if I'm going to use it that way. Things that are observable, testable, and repeatable. Science can only deal with the natural world. And if you want to be real particular, things that can only be observed, tested, and repeated. Science can never be 100% sure of anything because the best theory in the world can be disproven, at least in some way, in some situation tomorrow. And then science also can't deal with the supernatural. It's confined to the natural world. So whatever you believe about ghosts and miracles and gods and creative beings, whatever, that is outside the realm of science. We can think about it kind of scientifically, but it's beyond the realm of science. All right, why is a class like this important for anyone, including non-science majors in college? I like to use the term biologically literate. You should have a basic understanding and maybe more importantly, an appreciation of you, all living things, and the world around you. I don't read fiction. Like, to me, learning about us, our amazing bodies, and the world around us is the coolest thing there is to do. Now, I'm clearly biased and I'm a geek, but I don't care what Harry Potter's doing. I did read the Game of Thrones, I have to admit, but I don't care what Harry Potter's up to because learning about the things my liver is doing right now is amazing. So I want you to leave this class with an appreciation of the world around you, and if you do that, I'm happy. All right, chapter 2. There'll be a little more just detail-based stuff here. What is an atom? What are the three subatomic particles, and where are they located in an atom? So an atom is the basic unit of life. If you look at the periodic table, what you see is atoms. The smallest amount of an element that maintains the properties of that element is an atom. So what are the three subatomic particles? We have protons, neutrons, and electrons. Here's how I remember them. Protons, positive charge, pp. Neutrons are neutral. They don't have a charge, NN. So protons are positive, neutrons are neutral, and electrons have a negative charge. So where are they located? Protons and neutrons are in the nucleus of an atom. And you have content videos to cover all this. Electrons are in a cloud on the outside of the nucleus. Do all subatomic particles weigh the same? No. Protons and neutrons are actually pretty similar, but they're almost all the weight. They're almost all the weight of an atom is protons plus neutrons. That's going to be the atomic mass of an atom, is its protons plus its neutrons, because it would take something like 2,000 electrons to equal the weight of one proton. So the answer to that question is no, because electrons are tiny. All right, what is an isotope? Why are they potentially dangerous? Why are they potentially useful? And how are those two things connected? So an isotope is when you take an element. So to be an element, the number of protons determines which element you are. The number of neutrons determines whether or not you're a neutral element or an isotope. So an isotope is an element with a different number of neutrons. So like example would be carbon dating. We have carbon 14, carbon 13, carbon 12. They have different numbers of neutrons. So why are they potentially dangerous? Well, they're unstable. So isotopes are radioactive. So of course we know that radiation can hurt us and kill us. Why are they potentially useful? Because they're radioactive, though. So we use carbon dating. We can use this little bits of radiation, but really because of radiation therapy for cancer. So isotopes are used for cancer treatment. So why they're dangerous and why they're useful, same thing. They're radioactive. The instability is what gives them their power. Look at things like uranium and plutonium. They power nuclear. They're so unstable. They give off massive amounts of energy, and that's what powers a nuclear power plant. A nuclear power plant is a steam-driven turbine system with one really dangerous room. The nuclear material is being used to superheat water, and then the steam that's created spins turbines and generates massive amounts of energy. So they're what makes them dangerous and what makes them useful are actually the same thing, the fact that they're radioactive. Okay, what is the difference between an ionic bond and a covalent bond? If you want to look at this real simply, an ionic bond is created when an electron is taken from one atom and given to another, a transfer of electrons. Covalent bonds, they're sharing an electron. So those are the key words. Ionic bonds involve a transfer of electrons. Covalent bonds, sharing electrons. What is an electrolyte? Why they matter to human health? Electrolytes are ions. So ions in the body are called electrolytes. So you're probably thinking Gatorade right now, something like that. And that's true. Sodium and chloride and potassium, these kind of things, these in their ionic form, they're called electrolytes in the human body. They matter to human health because we absolutely can't survive without them. Sodium, potassium, great. I'll just give you one good example. Sodium and potassium are used to control where fluid is in your body. So the amount of potassium inside your cells determines if they're full of water or not. The amount of sodium on the outside determines if the water escapes your cells or not. So if you eat a low potassium, high sodium diet, water's going to leak out of your cells. So your cells are going to be shriveled up and not be as healthy as they should. And you're going to be carrying too much water between your cells, which is what's called the interstitial fluid. This is why people that eat a high salt diet might have a puffy face, swollen ankles, high blood pressure, these kind of things. So they're very clinically significant. All right, know some key facts about water. Does water behave like you might expect it to based on its shape and weight? If not, why does it misbehave? So let's start there. It does not behave. Water should boil around like negative 83 degrees. And of course, it boils at 100 degrees Celsius. That's because of hydrogen bonds. So hydrogen bonds, these special kind of covalent bonds. So water doesn't behave like it should. And if it did, life wouldn't exist. On the earth, all water would be in the gaseous state. Ice floats, right? That's a cool thing. Solid water is less dense than liquid water, which is why ice floats. That's cool in your glass. But the reason that's important is our oceans would freeze from the bottom up and they'd be solid. Like during the summer, maybe we'd get like 3 feet of water on the top. There would be no aquatic organisms, no aquatic ecosystems. So water has a real special ability to hold heat. That's what keeps the planet from being like 200 degrees versus negative 200 degrees. It has a cohesive nature, so water can actually be pulled up plants and trees and these kind of things. All these things don't seem like huge deals, but life wouldn't exist without all these things. If water didn't have this cohesive nature, you can set a pin on water. Certain bugs and stuff can walk on water. If that didn't happen, well, we couldn't have plants that were off the ground at all. Or if ice didn't float, we wouldn't have aquatic ecosystems. We wouldn't have any ecosystems. And if water didn't have this high heat capacity, the planet would be too hot and too cold to live on. Like these are all unbelievable things. So water's very special. Watch the video on the life-giving properties of water. And the reason it is so special is because of those special bonds called hydrogen bonds. All right, understand the parts of the equation. Solution equals solvent plus one or more solutes. So a solution is a solvent plus solutes. But a solvent, so what's a solvent and what's solutes? Well, there's only one solvent, and that's going to be what's in the highest concentration. And then there can be one or more solutes. Those are going to be lower concentrations. So the example I always use is Kool-Aid. So a solution would be your finished Kool-Aid. I like the green stuff, whatever it was. The solvent will be the water, unless you chew your Kool-Aid. Water is going to be there in the highest concentration, so it's going to be the solvent. And water is called the universal solvent in the human body. And then one or more solutes. The solutes would be the Kool-Aid powder, packet, whatever, and the sugar. Lots of sugar, right? So that's what solutes are. But metals can be solutions. Air is a solution. So air, it's not about importance. The solvent in air is nitrogen because it's there in the highest concentration. It's useless to us. We don't use nitrogen that way. We use it. We get it from eating protein, but we can't use atmospheric nitrogen. So the solutes in air would be the oxygen, carbon dioxide, water vapor, those kind of things. So pretty cool. All right. What does the pH scale measure? Why is it called a logarithmic scale? So pH is basically talking about like concentration of hydrogen ions. So the pH scale goes from 0 to 14. 7 is neutral, where the number of hydrogen ions, H plus, and the number of hydroxide ions, OH minus, would be the same. And as you climb above 7, that's a base. It has less hydrogen ions than something that's neutral. If you climb below 7, it's going to be more acidic. So acids have more hydrogen ions, bases have less. But it's a logarithmic scale because every shift down or up is a 10x change in hydrogen ion concentration. So something with a pH of 6 has 10 times more hydrogen ions than something with a pH of 7. Something with a pH of 5 would have 100 times more hydrogen ions than something with a pH of 7. So 10 times 10. So it's huge differences. All right. What is an acid? Acids are going to donate hydrogen ions. That's why they lower pH, because now there's more hydrogen ions around. So they donate hydrogen ions to lower pH. Bases accept hydrogen ions. They remove them, which causes the pH to go up. And then buffers are anything that resists changes to pH. If it weren't for buffers, we would all be dead every second of every day. Like buffers are what keep your blood pH stable in an ever-changing world. So buffers is anything that resists change to pH. So what a buffer does is it acts like a weak acid in the presence of bases, and it acts like a weak base in the presence of acids. So that's why they work. All right, know the basic introductory information about carbohydrates, lipids, and proteins. Lipids are fats. Basically solid versus liquid. They will be covered in more detail later, so a 10,000-foot view is fine for now. So just real big picture, carbohydrates, think of them as an energy source. You don't get a lot of energy from carbohydrates, from the ones that we use for energy anyhow, like sugar, but they're quick sources of energy. So carbohydrates, think energy sources. You've got, we'll cover the simple versus complex carbs in the next question. Lipids, also think energy sources. More dense. You can get a lot more energy from lipids, but they burn a lot more slowly. But lipids also play a role in insulation. A lot of your, some of your hormones are made of lipids, these kind of things. And then protein. So I always like to say, think of carbs and lipids as energy sources first. Do they have other functions? Sure, but they're primarily energy sources. Think of proteins as building blocks. You can use protein as a food source, or calories and energy, but they're more of a building block, and I'll explain why in a later question. All right, so carbs, quick energy, lipids, more energy but burn real slow, and proteins, think building blocks. Proteins do things that only proteins can do. Another way to look at that, let's see. So carbs, there's no such thing as an essential carbohydrate because your body can make them. Lipids, there are essential fatty acids. You don't have to eat saturated fats. You don't have to eat the monounsaturated fats like olive oil and stuff, but there are some essential fatty acids. And proteins are essential too. We have essential amino acids. There are things, which I'll get into in a moment, that only protein can do. All right, what is the difference between a simple sugar and a complex carbohydrate? So it depends on size. Your monosaccharides like glucose, your disaccharides, mono meaning 1, di meaning 2, these are small. So like sucrose, which is table sugar, lactose, which is milk sugar, those are simple sugars. Your complex carbohydrates, those are going to be starch, long chains of glucose, and cellulose, which is, fiber, but still long chains of glucose, we just can't digest it. And the third one is glycogen. That's how your body stores glucose, in long chains. So the difference is size. Small carbohydrates are going to be simple sugars. Large carbohydrates are going to be complex carbs. All right, what makes glucose such a good energy source? Are there any downsides? It is quickly burned for fuel, and it's generally, we all have it. Even if you're on a low-carb diet, your blood sugar is still elevated or normal. So it's a quick energy source, but its weakness is you don't get a lot of energy from it. So fat, I like to think of it this way. Fat would be logs on a fire. You're going to get tons of energy, but it doesn't burn quickly. If you want to start a fire, you use kindling, you use newspaper, this kind of stuff. That's carbs. Carbs are quick energy sources, but they don't give you a lot of energy in return. Fats are better energy sources, but they burn slowly. So that's why we burn both. We're basically flex fuel vehicles. What is the difference between a saturated and unsaturated fat? Can you tell us by looking at a fat that it is one or the other? So saturated fats, they're going to have no double bonds. That's why they're saturated. And they're going to have straight chains. Unsaturated fats are going to have one or more double bonds, and they're going to have kinks in the chain. So the reason, think about like stacking plates on each other versus stacking things that have really strange shapes. The plates will stack closer together. So you can, by looking, if a fat is a solid at room temperature, it's a saturated fat. Butter, lard, these kind of coconut oil. If a fat is liquid at room temperature, it's going to be an unsaturated fat. Olive oil, vegetable oil, etc. So it's because of the saturated and unsaturated fatty acids that they are solid or liquid at room temperature. All right, what element makes protein different than carbs or fat? And that is nitrogen. All three of them are made of carbon. They're organic molecules made of carbon, hydrogen, and oxygen. But protein also has nitrogen, the fourth most common element in your body. So that's why we say protein can do things that only protein can do, because we need it for our nitrogen. You have to consume protein. What are enzymes? What are they usually made of? What do they do? Why are they critically important to all living things? So enzymes are going to be catalysts. A catalyst, or I like to call them biological catalysts in this case. Catalyst is something that speeds up a chemical reaction without being changed by it or changing it. So it speeds up a reaction without actually changing it. So that's why they're really important. We'll come on in a second. What are they usually made of? Protein. Almost all enzymes are made of proteins. What do? So they speed up these chemical reactions. They do so by taking really random things that kind of would happen on their own if you gave them enough time and controlling them. So they happen with a lower activation energy. It takes less energy to turn reactant A into product B with an enzyme than it would without. So why is it important? Well, without enzymes, there's no life. I mean, even cosmologists, when they study what could the simplest cell be, I mean, it's going to need hundreds, if not thousands, of enzymes. Because our metabolic reactions would occur so slowly that we couldn't do it. We couldn't live. So that's why they're important. But here's why they're really important. There are other ways to speed up chemical reactions. In a lab, I can add fire, I can add heat, I can add acid. Well, if you want to go on a run, you can't drink battery acid. Your body temperature can't reach 140 degrees. So it's a way to speed up chemical reactions without having to destroy our ability to maintain homeostasis, that stable internal environment. All right, what does it mean when a protein is denatured? So you've all seen this when you cook an egg. So that clear, we call it egg white, but it's clear when it comes out. As you cook, as you increase the heat, the proteins that are in that egg white they'll denature. And as they unravel, they become less soluble in water and they thicken up. And that's you actually can watch that happen in an egg. So when proteins, all proteins, including enzymes, are sensitive to pH changes, temperature changes, etc. So that's why we have to maintain homeostasis. When you hear somebody talk about organ failure, like if somebody has a fever and they go into organ failure or something like that, what's really happening is their pH or temperature is changing, or something like that is changing so much that their enzymes are going to denature and unravel. So denaturing is when a protein unravels. And the reason that's important and why it's a bad thing is proteins are based on structure. So think of proteins as keys or locks. If you go home and your lock got so hot today, which it won't, but your lock got so hot today that it changed shape. Your key wouldn't work anymore, and you couldn't open that door. Well, that's what would happen if your proteins lose their shape. If a protein loses its appropriate three-dimensional shape, it loses its function. So if a protein denatures, it loses its shape, and then there goes the function. All right, know the basic structure of DNA. In what ways is it similar to RNA? In what ways is it different? All right, so DNA and RNA, they're both built from nucleic acids. They both have a sugar phosphate backbone. But here's the key differences. DNA is double-stranded. RNA is single-stranded. DNA is made of the building blocks A, C, G, and T. RNA is made of the building blocks A, C, G, and U. So you replace the T, thymine, with U, uracil, its cousin. So the videos go through this. And then and the third difference is DNA, that sugar phosphate backbone, the sugar is deoxyribose. That's why it's called DNA. And RNA, that sugar in the sugar phosphate backbone, is ribose. That's why it's called RNA. Let's see. What does ATP stand for and why is it important? ATP stands for adenosine triphosphate. Why is it important? That is the cellular energy current your body uses. We talk about using glucose for fuel or fat for fuel. No, we use those to make ATP. ATP is actually what powers your body, powers your metabolism. So your metabolism's job is to make ATP every second of every day. So that's what it is. I like to call it your cellular energy currency. All right, just kind of scanning around here. Number 3. What is a cell and what is the cell theory or cell doctrine, this textbook calls it? So a cell is the basic unit of life. The cell theory says that all living things are composed of cells. And number 2, all cells come from pre-existing cells. That's to be alive, you have to meet those qualifications. All right, what are features that all cells have? And what are features that eukaryotic cells have that prokaryotic cells do not? So all cells are going to have DNA. They're going to have a cell, a cell membrane, plasma membrane, and they're going to have a cytoplasm, the cell guts. They'll have all those things. But there are a lot of different differences. Different cells are going to have different structures. But so prokaryotes, it means pre-kernel or pre-nucleus, and eukaryote means true kernel or true nucleus. So eukaryotic cells are going to have a membrane-bound nucleus, whereas prokaryotes do not. Bacteria have DNA; they just don't have it's not stored in a nucleus. But the more technical definition would be that eukaryotes have membrane-bound organelles. So when you're learning about the organelles, that would be like your rough and smooth endoplasmic reticulum. It'd be the Golgi apparatus, those kind of things. Mitochondria. Those are all examples of organelles that bacteria don't have, that prokaryotes don't have. All right, why are the cells of a mouse and the cells of a blue whale the same size? Well, cells can't get any bigger. Like if you're going to be a bigger organism, like I'm a big guy, I'm made of more cells than you, not larger cells than you. But cells have a finite size they can get because as a cell gets bigger, the volume inside the cell gets too big for the surface area on the outside. What that basically means is if a blue whale had bigger cells than a mouse, it couldn't get nutrients to the center of that cell and it couldn't get waste products out. So a blue whale is bigger than a mouse because it has more cells, not larger cells. All right, what are the 3 most important types of microscopes? I only had you watch a video on the light microscope. That's going to be the microscope you work with in the lab. I put a whole playlist of videos on microscopes in the course shell. That's a light microscope. But when you're seeing really cool structures and images in a textbook, it's usually going to, or on the internet, it's usually going to be electron microscopes. So you have the light microscope, which can see things, give or take, at about 1,000 times magnification. You have the scanning electron microscope, which can see about 10,000x magnification, but it can see surface structures in 3 dimensions. So if you see like just really cool images, they're usually called SEM, scanning electron micrographs. Then we have the transmission electron microscope. That can see at 100,000x or above, and it looks at internal structures. So that's the answer there. Know all the key cell organelles. I'm not going to walk you through them right here. I had you watch a 15-minute video that goes through those, but I picked a few to talk about here. So make sure you've done your rest of your homework. You need to know all your organelles. What's the difference between rough and smooth endoplasmic reticulum? So they're both these channels that materials go through, but the rough ones are rough because they're studded with ribosomes. They have ribosomes on their surface, and ribosomes are involved in protein synthesis. So when you think rough ER, think ribosomes, so think proteins. They modify and package proteins. Smooth ER, smooth endoplasmic reticulum, is smooth because there aren't any ribosomes. So don't think proteins at all. The smooth ER is used to make fats and sugars and also detoxify drugs and toxins. Next, what's the difference between a free and a fixed ribosome? So they both synthesize proteins. The best way to look at it, free ribosomes are just floating around inside the cell. They make proteins that are going to be used by that cell. Fixed ribosomes are on the surface of the rough ER, so they're going to make proteins that leave the cell and go somewhere else. What's the difference between a lysosome and peroxisome? They're both used to detoxify things, but lysosomes basically use digestive enzymes and acids. They break down old materials to recycle them. Peroxisomes can neutralize things like alcohol and free radicals and stuff. So lysosomes think digesting cell parts, that kind of stuff. Peroxisomes, they're going to be primarily involved in detoxifying lipid-based things, alcohol, that kind of stuff. What are the 3 ways that the plasma membrane or key ways, sorry, there aren't 3. What are the key ways the plasma membrane or cell membrane are described? So you're going to hear it called the phospholipid bilayer, and you can see the pictures of it and watch the videos about it. It's a bilayer, double layer of phospholipids, which have a phosphate head and these 2 lipid tails on each of the phospholipids. That's the basic shape. But you're also going to hear it called a fluid mosaic model. It's called a mosaic. I think about like a picture made of 1,000 pictures, right? So a mosaic, because it's made of multiple ingredients. There's not just phospholipids. There's carbs there, there's other fats there, there's cholesterol there, there's proteins there. It's called a fluid mosaic model because it's not rigid. You see these pictures and you think it's rigid, but no, everything is free floating, kind of like some stuff floating on a pond. Everything's free floating through your cell membrane. You would totally think it'd be rigid, your cells would be rigid, but the consistency of a cell membrane is about the same as olive oil. They're not near as tough as you'd imagine, so kind of interesting. All right, what is the often misunderstood ingredient in plasma membranes that makes them strong yet flexible? That is cholesterol. We are terrified of cholesterol, but every cell in your body makes cholesterol because every cell in your body needs it. Without cholesterol, your cells would be tearing open. All right, and they also wouldn't be, they'd be too stiff. What are the key ways materials move in and out of cells? So the big ones I focus on are diffusion, osmosis, and active transport. So they all actually very similar definitions. Diffusion is the movement of solutes from an area of high to low concentration. Osmosis is the movement of water, which is the solvent, from high to low concentration. And then active transport is things are moving from low to high concentration. So what's the key difference between diffusion and osmosis? In diffusion, the solute is moving. So like you put a tea bag in tea, you're in water, right? The tea is going to escape the bag and spread throughout the water. That's the solute. The tea is moving, not the water. Osmosis is, the simplest way to define osmosis is water following solutes. So the water is going to move where solutes can't. So that's going to be, so osmosis is considered the movement of water across a semi-permeable or selectively permeable membrane. But I like to just call it the diffusion of water. Water is moving from a high concentration to low concentration. But the simplest, the best definition to me of osmosis is water following solutes. When you sweat, salt goes to the surface of your skin and water follows it out. Water just following solutes around is what osmosis is. Next, what's the key difference between passive and active transport? Passive transport doesn't require energy. Active transport does, because so you have to power active transport. So active transport requires ATP. Passive transport does not. So active transport, the best example of the book, well, that's actually the next one. What's the key example of an active transport system in the human body? It's called the sodium-potassium exchange pump. It takes massive amounts of energy to power this pump. We have to constantly pump sodium out of our cells and potassium into our cells. So why is that important? It maintains cell volume, but it also, your nerves wouldn't work without it. So your brain is like 2 or 3 pounds. It's a fraction of your body weight, but it consumes 20 to 25% of your energy. 40% of the energy your brain's going to use today is powering the sodium-potassium exchange pump. Pretty amazing. All right, so that's a good example of an active transport system. What happens to a cell that is put in an isotonic solution, a hypertonic solution, or a hypotonic solution? So isotonic means equal tone, equal concentration. That means there'll be balance. If you put a cell in an isotonic solution, it won't change shape. The same water moves. We can't stop water from moving, but there'll be no net change in water or no net movement of water, because every time a water molecule enters the cell, another one's leaving. So that's an isotonic solution. In a hypertonic solution, which is very common, if people, we consume too much sodium, not enough potassium, not enough water, these kind of things. A hypertonic solution, remember, there's going to be more solute outside the cell. Osmosis says water is going to follow solute. So what happens there is water is going to leave the cell. The cell is going to shrivel up, and there's going to be more fluid on the outside. In a hypotonic solution, that's the opposite. There's less solute outside the cell, so water is going to rush into the cell, and that cell is going to swell. So quick answers: isotonic, no change. Hypertonic, the cell is going to shrivel up. Hypotonic, the cell is going to swell and expand. Understand the parts of the equation. Metabolism equals anabolism plus catabolism. Your metabolism is the sum of all the chemical reactions and physical processes that occur in your body, but it's the combination of these two things. Anabolism is building up, things that require energy to cause growth. Catabolism, you break things down to release energy. So your metabolism is the sum of all the things you spend energy on building and release energy from when you break it down. So think about it this way. You eat. You digest your food. That releases energy in the form of the calories that your body's going to use. Then you take that energy and you take those building blocks to build you. So if you had some bacon this morning, the protein in that bacon is going to be in your leg muscle later today. So that's what your metabolism is there. All right, I will only ask you to understand cellular respiration at the introductory level. Use the metabolism fact sheet as your guide. Really just what's like the main thing that happens during glycolysis, what's the main thing that happens during the Krebs citric acid cycle, what's the main thing that happens during electron transport system. I'm not going to go into like the videos, the videos that are there. I mean, you can spend an hour easily just watching videos on metabolism. Just know the basics and you'll be totally fine. All right, what is needed for cellular respiration to occur and what products exit the reactions? So cellular respiration is, we need oxygen, right? That's why we have to have oxygen. So here's the equation. Glucose or whatever fuel source we're using, glucose would be C6H12O6. It's on your fact sheet that I gave you. Glucose plus oxygen, you have to have oxygen, will lead to ATP plus waste products, carbon dioxide plus water. So glucose and oxygen, or some fuel source and oxygen, will lead to carbon dioxide, water, and energy in the form of ATP. All right. Next, you will notice that throughout the cellular respiration pathways, electrons are very important, but in the end, the movement of electrons is not what creates energy in the form of ATP. What does? It's actually the movement of protons. So if you watch those videos and you go through your metabolism stuff, we take electrons, we carry them, and we release them. The pumping or movement of electrons causes protons to be pumped outside of the mitochondria. And as they come back in, that's actually what creates ATP. So I won't go into more detail there because you don't really have to. But why is a hydrogen ion and a proton the same thing? So it's a good point. So an atom should have protons, neutrons, and electrons, but hydrogen does not. Hydrogen is the only atom that doesn't have neutrons. So a hydrogen ion, what that means is you've removed the electron. So with hydrogen on the periodic table, you started with a proton and electron. You removed the electron. The only thing left is the proton. So some sources will talk about hydrogen ions with metabolism. Others talk about protons. It is the same thing. I just wanted to clear up that little thing there. All right. Chapter 4. Define a tissue and define an organ. So we've already done this. A tissue is when you take a group of cells that are working together with a similar function. So you take like a cardiac muscle cell. You put all the cardiac muscle cells together. We now have cardiac or heart muscle tissue. Define an organ, 2 or more tissues working together. So when you add the electrical conducting system to the heart muscle tissue and you add the fibrous skeleton of the heart, now we have a heart. So that's a tissue and an organ. What are the 4 tissue types in the human body? So every tissue in the body is either going to be epithelial tissue, connective tissue, muscle tissue, or neural tissue. So real quickly, epithelial tissues, they line and cover things. So they line cavities and they cover any passageway in and out of the body. I'm going to skip connective tissue. Muscle tissue contracts and gets shorter. Think muscles. And neural tissue, nervous tissue, sends and receives electrical impulses. The reason I skip connective tissue is because everything else is connective tissue. So if it's not lining or covering something, and it's not contracting, and it's not sending and receiving electrical impulses, then it's connective tissue. So it's a catch-all, really. All right. Where will you find epithelial tissues? And what are the key ways to classify them? So again, lining cavities and any passageway in and out of the body. So think glands, your GI tract, reproductive tract, etc. How do you classify them? Simple, there's a video about this, but simple versus stratified. So if they're simple, they're a single layer. If they're stratified, they're multiple layers. And then we have squamous, which means they're flattened like egg-shaped cells. We have cuboidal, they're cube box-shaped cells. Or columnar, they're shaped like columns. So simple squamous is going to be one layer of these egg-shaped cells. Stratified columnar would be multiple layers of these column-shaped cells. So next question, what are the simple epithelium generally good at? They're going to be really thin and delicate, so definitely not protection. They're good at absorption and secretion. They're good at allowing things to move through them. So what are stratified epithelium good at? They're tougher, thick layers. They're protection. So whenever you see a stratified epithelium, think protection. A simple epithelium, think absorption and or secretion. How could blood, bone, and fat all be classified as the same tissue type? That's because to be a connective tissue, all you have to have is specialized cells, which all 3 of those do. You have to have a fluid ground substance. So there's going to be a lot more fluid in blood than bone, but they both do have a ground substance. And you're going to have proteins. So blood has plasma proteins. Bone is like 30% protein. So really, that's why connective tissue is this big catch-all, because all the tissue types, except for the 3 I already mentioned, are going to fall in that same category. So they're all connective tissues. You may not think of blood as connective tissue, but it is. It's a fluid connective tissue. All right, what are the 3 types of muscle tissue? What are the key characteristics of each type? So there's a video on this, but we have skeletal muscle, cardiac muscle, and smooth muscle. So skeletal muscle is voluntary. It's the only one that is, which means you control it. I'm wiggling my fingers right now. You can't see it, but it's because of skeletal muscle. So skeletal muscle is voluntary, whereas smooth muscle, so skeletal muscle is the muscles you think of. Smooth muscle is going to be lining all your hollow organs, including your blood vessels and GI tract, etc. And cardiac muscle is going to be in your heart. So skeletal muscle is voluntary. You control it. Smooth muscle is involuntary. You don't control it. It's subconscious. And cardiac muscle is also involuntary. The next way to classify it is how many nuclei, or how many nuclei they have. Skeletal muscle has multiple nuclei. There's tons of them. It's because a skeletal muscle cell is really a bunch of cells that fuse together, whereas smooth muscle and cardiac muscle only have a single nucleus. And the third thing is that skeletal muscle is striated. It's an organized banded appearance. Skeletal muscle is striated. Cardiac muscle is striated, but smooth muscle is not. They're more like spindle, like lip-shaped cells, basically. They're not near as organized. The only other thing you might need to know, this will be later, but cardiac muscle cells also have special electrical connections. They're called intercalated discs. I'll just tell you the word now. Just you don't have to know it yet, but you'll hear it later. All right. What are the key features of each of the 11, or some would say 12 organ systems? The reason I say that is because I like to separate the male and female reproductive systems. They're so different. So that if you do that, there's 12. But every human has 11 organ systems. Key features, again, I won't walk through them all right now. You have organ system. Make sure you've watched the organ system review videos. It's a two-parter where I do give you the key features of each one. Next question then, what is a membrane? A membrane is going to be epithelial tissue plus the connective tissue below it. They oftentimes call it the basement membrane. So membranes are going to be epithelium plus the underlying connective tissue. So like your skin is called a cutaneous membrane. All pathways in and out of the body are going to be lined with mucous membranes. Those would be some examples of your joints, your synovial joints, like your knee and elbow. They're going to be lined with a synovial membrane. So those are going to be, and then you have your serous membranes too. But those would be examples of membranes. All right, how is your body's method of maintaining homeostasis similar to the central air unit in your home? So we have your body uses receptors to monitor what's going on, just like your central air unit uses a thermometer to know what the temperature is in the house. It has a control center. The thermostat in your house would be usually your brain or spinal cord for your body, and it has effectors. So if the temperature in your house is climbing and you have the thermostat set at 70 degrees, and the receptor tells the thermostat, hey, it's 71 degrees in the house, it's going to do something. It's going to turn the air conditioner on and bring the temperature back down to 70. Well, if your core temperature is 98.6 degrees and it starts climbing, your temperature receptors, called thermoreceptors, are going to tell your brain the temperature is climbing in your body, and it's going to do things to lower the temperature. The same system. So that's why they're similar. What is negative feedback? What is positive feedback? Almost all body processes are controlled by negative feedback because, remember, it's hard to be alive. You already have homeostasis, or should, so your body's already maintaining a stable internal environment. If something changes, chances are it's bad. So let me give you an example. I just did the body temperature one. So if your body temperature goes up and your body says, hey, our body temperature is going up, let's make it go even higher, that'd be terrible. We would die. Negative feedback says if there's a change, it's almost always bad. Let's get rid of it. So our body temperature goes up. We respond by bringing it back down. That's negative feedback. So negative feedback, if there's a change, your body tries to negate that change or fix it. Positive feedback, think about a snowball rolling down a hill. As if there's a change, your body says, we need more of this change. So that's going to be pretty rare. We don't want that to happen very often, but think about this example. You start bleeding. So the blood clotting cascade is a positive feedback system. What if your body says, okay, you're bleeding, your blood is starting to clot, the wound is starting to clot, let's stop that. That'd be a bad idea. Your body says, hey, let's get this blood to clot as fast as we can. As we'll learn later, the blood clotting cascade makes your blood clot 1,000,000 times faster than it would without the cascade that we have. So same thing with childbirth. So those will be the two examples on the next one. Why are almost all body processes controlled by negative feedback? Because our body is already in a state of homeostasis. We don't want it to change very often. The two key exceptions would be labor and delivery. Once that starts, you want to speed that process up. You do not want you don't want your body to stop having a baby once it starts. You don't want your blood to stop clotting once it starts. So those are the two key exceptions of positive feedback, labor and delivery and blood clotting. And so think emergencies. In emergencies, your body will use positive feedback when something has to happen quickly. All right, that's an hour-long review. I know it's long, but I covered a ton of important stuff there. And in an online course, especially for this first unit, or I'll do it for all the units, but I don't get to lecture to you guys very often, and this is kind of how I do it. So I know you only know now at the end it was an hour long, but I certainly think it'll be worth your time. So I wish you all the best of luck on your Unit 1 exam. I'll see you for the Unit 2 review.